Okay, imagine you're a doctor, right? And you are trying to find a single microscopic mutated cell hiding among literally millions of healthy ones. Or picture trying to detect a viral infection in a patient when there are only a handful of virus particles circulating in their entire bloodstream. I mean, it is the ultimate microscopic needle in a haystack problem. So how do we find something so incredibly rare, yet potentially so deadly? Well, welcome to this explainer, where we're going to unpack the absolutely fascinating technology that makes solving this impossible medical mystery a reality.
Today, we are diving into the DNA amplification revolution. We're going to take a journey from the invention of what was essentially the first biological photocopier all the way to the life-saving, absolute precision of modern digital PCR. It's just this incredible story of how scaling down to the microscopic level has allowed us to see things that were previously completely invisible. So, here's our agenda for today. First, what is DNA amplification? Second, the PCR recipe. Third, the limits of standard PCR. Fourth, the digital PCR revolution. And finally, saving lives with dPCR. All right, let's jump right into Section 1. What is DNA amplification?
To really understand the digital revolution we have today, we kind of have to anchor back to 1983. That's when a scientist named Kary Mullis developed polymerase chain reaction, or PCR. This was an absolute game changer. It changed molecular biology forever because it allowed us to take undetectable traces of target DNA and amplify them into massive, analyzable quantities.
But here's the thing, PCR doesn't just add DNA, it multiplies it exponentially. The formula is 2 to the power of n, where n is the number of cycles. So after 1 cycle, you have 2 copies. After 2 cycles, you have 4. After 3, you have 8. Because of this compounding exponential growth, just one single target sequence of DNA can literally be turned into billions of copies in a couple of hours in a lab. It's basically an exponentially progressing synthesis happening right there in vitro.
Moving on to Section 2, the PCR recipe.
To make this molecular amplification actually work, we need a very specific assay toolkit. Think of it like a recipe. First, you need the target DNA template. Then 2 DNA primers that act as start tags. Next is the real workhorse of the whole operation, DNA polymerase, specifically Taq polymerase. And fun fact, Taq survives the extreme heat of PCR because it was originally isolated from hot spring bacteria. Finally, you need your DNTPs, a buffer solution, and mineral cofactors like magnesium. Now, those DNTPs I just mentioned, those consist of the 4 basic nucleotides: adenine, thymine, cytosine, and guanine. These are the literal physical building blocks that the Taq polymerase enzyme uses to synthesize the brand new DNA strands. Without them, the whole amplification process just stops entirely. So you mix all these components into a tube and you pop it into a thermal cycler. The entire basis of PCR is completely reliant on rapid temperature changes. Step 1 is denaturation at a scorching 95 degrees Celsius. This literally melts the double-stranded DNA apart into 2 single strands. Step 2 is annealing, where the machine cools down to around 40 to 65 degrees, which lets the primers bind to their specific target sequences. And finally, Step 3 is extension at 72 degrees. This is where that heat-loving Taq polymerase really goes to work, extending the chain by adding those DNTP building blocks. You repeat this cycle 20 to 40 times, and boom, billions of copies. Okay, Section 3, the limits of standard PCR. So, the crucial point here is understanding the difference between standard quantitative PCR, or qPCR, and digital PCR. For the past 20 years or so, qPCR has been the gold standard. But if we're being honest, it's an analog technology. It relies on standard curves for calibration, and the operator often has to subjectively choose a signal threshold. Because of this, standard qPCR is kind of like guesstimating a recipe. Even in highly trained labs, you can see a 20 to 30% variation in the results. Digital PCR, on the other hand, eliminates that standard curve completely. It uses Poisson statistics to count the exact absolute number of molecules. No guesstimating at all, just absolute quantification.
Which brings us to Section 4, the digital PCR revolution.
The true breakthrough here lies in partitioning. See, standard qPCR conducts amplification in one single bulk tube. Digital PCR physically partitions that exact same sample into hundreds, thousands, or even millions of tiny, separate compartments. You dilute the sample so much that every single microscopic compartment ends up with either exactly 1 target DNA molecule or 0. You aren't measuring a glow and guessing the concentration anymore, you are counting individual molecules 0 or 1, just like binary digital code.
To really grasp the sheer scale of this digital revolution, consider this number: 1 million. Modern commercial platforms, like the Raindance digital PCR system, can take a single standard sample and partition it into 1 million distinct picoliter water and oil droplets. Literally, every single droplet acts as its own independent PCR reaction. The engineering required to do this reliably is staggering, and for sure, it completely changes the game for accuracy.
Let's bring it back to the real world in Section 5, Saving Lives with dPCR. Because in a clinical setting, absolute precision matters. Standard PCR actually struggles to detect very low viral loads, often missing infections that are below 50 copies per milliliter. But because digital PCR physically isolates the sequences, it completely removes the competition for chemical reagents. This allows doctors to accurately track very low-level HIV viremia, which can actually predict if a patient's antiviral therapy is failing long before traditional tests ever catch it. It also lets us quantify occult, or hidden, viruses like GBVC. But honestly, perhaps the most exciting clinical impact is in oncology.
Think about it like this. In a bulk PCR reaction, if you have one mutated cancer gene floating in a sea of healthy DNA, the healthy DNA just hogs all the PCR ingredients, and the mutation stays totally invisible. But when we compartmentalize everything into droplets, that single mutant gene gets isolated into its own little droplet. Suddenly, it has no competition. Researchers have proven that using this method, they can confidently detect just one mutant KRAS cancer gene hiding amongst 200,000 healthy genes. That represents a 0.005% detection rate. It is an absolute breakthrough for early cancer detection and targeted therapy.
We've moved from a messy analog guesstimation to absolute single-molecule digital precision. By simply taking a sample and dividing it into millions of microscopic drops, we can now find the ultimate needle in a haystack. Which leaves us with one incredibly exciting question to think about. What other invisible medical mysteries will this digital partitioning technology unlock next? Well, thank you so much for joining me for this explainer. I really hope this deep dive into the world of DNA amplification has sparked your curiosity as much as it has mine. Keep learning, and I'll see you next time.